Hello everyone, my name is Marilina de Sauto. I am a PhD student from the University of Buenos Aires and I'm going to talk about the plasma membrane calcium ATPase activity is modulated by aluminum due to its differential interaction with lipid bilayers. The PNCA is a PATPase that its function is to transport calcium from the cytoplasm to extracellular medium against its concentration gradient hydrolyzing ATP. This protein is located in the plasma membrane of all eukaryotic cells and is regulated by cellular mechanisms that some of them are being studied. Like other ATPases, this protein goes to different conformations to transport the calcium out of the cell. Here I show you the sarcoplasmic calcium ATPase CERCA that its structure has been resolved in different conformations of the reaction cycle. The PNCA has not been crystallized yet. The structure has been resolved in the E1 conformation in 2018, but unfortunately the main original parts of the protein that are different from other PATPases have not been solved in this. In previous work, we studied the effect of aluminum on the pump, and we saw that aluminum can inhibit the pump with an 8 micromolar apparent constant, and calcium and magnesium do not protect from this effect. By using radioactive ATP, we saw that it can fix the pump in the phosphorylated conformation, and also with accessibility to proteases and kinetic experiments like the phosphorylation with ADP, we postulate that the pump is fixed in the E2P conformation. It is known that the pump is modulated directly by acidic lipids but also by the lipids in the surrounding bilayer. So we want to know if the effect of the aluminum in the pump can be due to its effects in the biophysical properties of the membrane. This is important since aluminum is related to neurodegenerative disorders, so it can affect another membrane proteins in the cells too. First, we measure the calcium ATPase activity as a function of the molar fraction of dimeristoyl phosphocholine by increasing the concentration of the lipid and decreasing the detergent. In the absence of aluminum, the activity of the protein has a biphasic behavior like it was described by Pignataro and collaborators. The protein is not active in the absence of lipid. As we increase the lipids, they can intercalate around the protein and this favors the activity. Then the activity decreases. This could be due to a change in the organization of the lipids in solution. In the presence of increasing concentration of aluminum, we saw two types of effects. At lower lipid concentration, the aluminum can inhibit the pump as we increase the concentration, but at higher lipid concentration, we saw a delay in the decay of the activity due to the presence of the aluminum. To study what was happening here, we performed such experiment in collaboration with the Professor Rosangela Itri and Gustavo Scarnacchi from the University of São Paulo. In absence of aluminum, we noticed that definitely we have two types of scattering curves at low or high molar fractions. By fitting these curves using ShemFit software, taking into account the electronic density and the thickness, we conclude that at low molar fractions, the molecules organize like a ferrocylinder structures, and the change in the thickness could explain the increase in the activity. As we increase the lipids, we have a transition in the organization to a big bilayer. In this, the thickness does not change too much, but we perform fluorescence experiments using Lordan probe, and we saw an increase in the generalized polarization function that indicates a less polar environment, and this could be due to a more packed membrane. That could explain the decrease in the activity because this change increased the lateral pressure in the protein and declined the turnover of the PNCA. We performed the same experiment in the presence of aluminum and we obtained the scattering curve of aluminum alone in solution. If we take the curves in the absence and we add them with the scattering curve of aluminum alone, we obtain the red curves, that they are very different from the one obtained in the presence of aluminum in solution, indicating that we have an effect on the bilayers and this effect is greater the more lipids we have. I wanted to thank very much the organizers, my advisors, especially Irene, the people of the lab, the collaborators. They have been all very important in this work, not only professionally, but also they have been supportive and kind, and I hope to see you soon.